Okay, well, thank you very much. So I want to give you a snapshot of where we are at Stanford Cardiovascular Institute's efforts to treat heart disease with human embryonic-derived stem cells. And these projects, which I'll talk about, sort of a dual-pronged approach, are both benefited immeasurably by support from CERN. We'll talk more about that in a little bit. So we're certainly not talking an orphan disease. We're talking a huge, huge disease, the #1 cause of death in America, and there's 5 million Americans living with heart failure, and there's many, many cases every year. And the problem is that even if we've gotten better at treating heart disease, it's still the case that by the time you're in heart failure, 50% of these patients are going to die within 5 years, which is a worse survival rate than a lot of cancers, which most people don't realize. It's an enormous healthcare burden here. And in a sense, what happens is you start typically, let's say, with a heart attack, ischemic damage, and you've got a scar region, so you've lost healthy tissue. Under the pressure the heart is subjected to, that scar expands, the wall thins, the LV chamber gets distorted. Your heart often tries to compensate by getting bigger and trying to be stronger, not by making new myocytes, because although we now know that the heart can make a few new heart muscle cells, it's really not its mechanism. Instead, the heart just tries to make each of its surviving myocytes bigger. And that works for a little while, but eventually it stops being good enough and you go into heart failure. There's other ways of getting to heart failure. There's drug damage or viral problems, things like that. But the question is, what can we do to stop this, or even better yet, reverse this? So there are certainly some options right now, and we know heart transplants work. There's not a doubt about that, but we also know the big problem with heart transplants is that there just aren't enough available organs. We also know about lifelong immunosuppression. So there's some very promising technology, left ventricular assist devices, which essentially take over for the left ventricle. And these are really remarkable little devices that show a lot of promise, and they've gone from being something which were only being used to keep a patient alive long enough in case a heart came up, to maybe what we'd call a destination therapy. So something that can keep these patients alive maybe for the rest of their lives. And this requires some really advanced technology, and right now LVADs have the considerable disadvantage that they're not biological tissue, that there are wires that protrude through the skin, so it's a source of infection and that sort of thing. And that's something which may be overcomeable at some point, but it's not quite ready yet. There's been a lot of different attempts at cell therapy for heart disease, and you've heard about some of these things even at this meeting, and it ranges from things like skeletal muscle to bone marrow-derived cells to mesenchymal stem cells under a variety of different guises, and there's a lot of different cell therapy attempts out there which really are using MSCs, which have just been isolated by slightly different methodologies. There's a variety of other things, including what people might call cardiac stem cells. The evidence is pretty strong at this point that you can put in these cells, and you may get a little bit of bang for your buck. You may get a little bit of improvement of function over the long term, but it's pretty clear that none of these cells are becoming new cardiomyocytes, despite what was originally thought. And it's still being argued whether any of these cells are triggering, let's say, the proliferation of what might be endogenous stem cells, because the very existence of those endogenous stem cells is highly, highly controversial in the field. So what we can say, though, is that all these approaches are trying to take cells which are not cardiomyocytes and trying to put them in and get them maybe to become cardiomyocytes. So what we are doing differently is that we're starting with, in this case, embryonic stem cells, although we could very well do this with another type of pluripotent stem cell, the induced pluripotent stem cell. And we're not putting in undifferentiated cells and asking them to become cardiomyocytes. We are making the cardiomyocytes in vitro and putting those cells in. We're putting the final cell type in, which is a very different approach. So what we're talking about here is you would start potentially with the same batch of cells, which would be between 100 million to 1 billion total cells, which we would heavily characterize and have characterized for cardiomyocyte markers, so that we know the cells that we're making are primarily ventricular myocytes. And we perform a variety of electrophysiological and functional assays to make sure that these cells are bona fide cardiomyocytes. And we know that these are not necessarily 100% pure cells, but we don't think they have to be, because the other cells in these preparations are other normal constituents of the heart, which include fibroblasts and smooth muscle and endothelial cells. And of course, anybody who's working with pluripotent stem cells is rightfully concerned about the potential for the persistence of undifferentiated cells, which might cause tumors. Therefore, we have to have extremely sensitive assays for any residual undifferentiated cells, and potentially ways of removing them if they're still present. So when you have these cells, there's essentially two parallel things you can do, and we are doing both of them thanks to CERN's largesse. On the one hand, we're taking these cells as is, so it's a suspension of cells, really, and injecting it into the damaged ventricle, and we want these cells to repopulate the infarct and provide contractile force. And this has been supported by a $20 million CERM disease team. It was a $200 million disease team grant. And the other approach is to take pretty much the same material but do something different. So instead, we're making what you might consider a biological LVAD. We're making a chunk of heart muscle tissue, which includes the cardiomyocytes and the fibroblasts and other constituents. And once we've measured these things in vitro and shown they have the appropriate properties you'd expect of a piece of heart muscle tissue by a variety of assays, we then transplant that into animal models and show that these things can provide contractile force. And here we were working off a CERM ET3 grant, which we're very grateful for. So this is early stage work. Our team here, both of these projects are headed up by Joe Wu, who's the director of the Stanford Cardiovascular Institute, and he's an expert on cells and differentiation and imaging and transplant. And the disease team grant is, the co-PI in this case is Deepak Srivastava at Gladstone, who's very well known for a lot of his really transdifferentiation work and study of cardiomyocytes. And both of these projects are supported by Larry Couture at City of Hope, who has been just done, frankly, a remarkable job at scaling up production of these cells to the point where there's just no bottleneck on cell production. There's no limitation whatsoever for cardiomyocyte transplantation. And with regard to the early translation grant, we're also working with Kevin Healy at Berkeley and also Wolfram Zimmermann, who's our co-PI in Germany, who's sort of the expert in making what I'm describing as, we refer to them EHMs, engineered heart muscle. These are the biological LVADs. And I'm managing both these programs. And my background is that for 16 years, I was at Geron Corporation, and I was the senior director of stem cell biology. And among other things, I headed up the cardiomyocyte cardiovascular disease project. So I've done a lot of this work with regard to how you make the cells, the preclinical modeling, the characterizations, the assays, et cetera. This is territory I know pretty well. So we have an expert scientific advisory board, again, names you would probably know in this field, from Mahendra Rao, Gordon Keller, Chuck Murray, Lior Gepstein, Josh Hare, and Andre Zeyer. And the latter two in particular have a great deal of expertise in transplanting various types of cells for heart repair trials. So this is a very powerful source of knowledge for us to draw upon. And the Stanford Cardiovascular Institute itself is where the cardiovascular research at Stanford occurs, and it's we have a lot of people involved in this, over 500 Stanford basic scientists. We've got the graduate students, the clinicians. We have the animal facilities, both small and large. And we are really experts at small animal and large animal models, and we need to use both for these. We know a great deal about catheter delivery of these cells because we're not going to shoot these things into the circulation and hope they home to the infarct. We're going to deliver them to the infarct. There's a great deal of imaging which is required for this, and we use anything from bioluminescence to track modified cells in small animal models, which is great, a non-invasive way of tracking long-term persistence of cells and testing various treatments which might prolong that persistence, to things which are more appropriate for large animals and human clinical studies, anywhere from echo to MRI to PET-CT. And again, at Stanford, there's a great deal of clinical trial experience, too. All right. So, as I mentioned, it's not just that the pluripotent stem cell is a great source of material because you can expand it indefinitely. You can create a master cell bank, for example, take one vial, start with 5 million cells in this vial, expand it 5 to 8 passages, you come up with a 200 vial working cell bank. One vial from that, you can expand 10 passages and make a god-awful quantity of cells, basically. So again, the starting material is reproducible and infinitely expandable. And at this point, working along with some very talented postdoc in Joe Wu's lab, a guy named Paul Burridge, and some work that came out of Sean Palachek's lab, Larry and his group have been able to really, really create unlimited quantities, really, of cardiomyocytes, very high purity cells. It's completely GMP compatible. We're not using any growth factors. It's all small molecule based. It's much more controllable in that case. And we have proof of concept for fully chemically defined media and substrates. And what I showed you here was a beating patch of cardiomyocytes. You can get very high purity. In this case, it was about 86% troponin T positive cells. And they stain with the appropriate cardiac markers, and they're cardiomyocytes. There's not much more to be said about it than that. But in order to do this, we know that we have to go through a variety of different kind of models, and this is, again, something I'm very intimately familiar with from my Geron days. So we know we have to be able to study efficacy and what the cells do and how to get them in and where they go when you put them in, Clearly, we're very concerned about tumor studies, so we're devoting a great deal of resources to that. And there's the standard tox studies. And we know we need 2 types of animal models for this. And we'd love to be able to do it all in a rat, but of course we can't do that. But you can do a lot of long-term studies. You can do persistence and tumor studies and tox studies. But ultimately, you have to move to large animal models. And you have to be able to keep these cells alive for long periods of time to demonstrate safety and efficacy. And just how long you're going to have to keep them alive in a large animal model is something which I think FDA is struggling with, because I'll tell you, a couple of years ago when Jaron talked to them about this, they said, well, you need to have your xenotransplants persist in a large animal for 9 to 12 months. And we realized that it may be impossible, in which case you can't go to the clinic. So we're working on that. We're discussing that with them. And recently there's been more of a realization that no one really has a great way of, say, tolerizing large animals to human cells that would give you long-term persistence. So the question is, how long is good enough? And that's something that we're all working with to try to overcome. So basically, we're very early. We're year 1 here. We've got our development candidate. We know how to scale it up, and we're doing our comparability in small animal models. So year 2, we come up with our specs for our pilot lots, so we do our proof of principle long-term studies in small and large animals. In year 3, we manufacture and release the material and initiate the IND-enabling studies. And year 4, we would then manufacture and release the material for the actual first-in-human trial, and of course complete the IND-enabling studies. And the goal is then to file the IND for the disease team in 4 years. And things we have going for us are that, I mentioned the immune issues are pretty significant in terms of getting these things to survive long enough in our animal models. Well, the good news is that the Wu lab in particular has a great deal of expertise coming up with ways of preserving these stem cells and differentiated progeny of stem cells in these animal models. Here's just some very recent papers we've done on how to keep these things alive using some novel co-stimulatory blockades. And also, as I mentioned, we're very concerned about tumor assays. Well, the Wu lab has also been very intimately involved in studies where we're trying to identify ways of removing any residual undifferentiated stem cells using some very specific surface markers, for example, against undifferentiated cells. And there's definitely precedent for the depletion of concerning cells from large lots of cells. Again, something I did at Jaron all the time. So we have the expertise to do this. We know how to do this. So just to summarize, we have inexhaustible material for these trials, and that's, again, thanks to the collaboration with Larry Couture at City of Hope and his group. So we have an unlimited supply of both the raw material and the actual cardiomyocytes made by a highly efficient GMP-compatible process. CERM has been instrumental in our efforts to generate both the suspension of cells and also the engineered heart muscle for both the disease team grant, where we're targeting the IND in 2017, and the early translation grant to make those sort of biological LVADs. We have a very experienced leadership team. We know generation of stem cells. We know how to differentiate them. We've got the animal models. Thanks to Larry Couture, we have the GMP cell production down. I certainly have experience with the industrial sides of cell therapy, and there's plenty of expertise on cell therapy trials. So we are very early stage, but I encourage you to watch this space because I think we're going to make a lot of serious progress on this problem. And with that, I'll stop. Thanks.